Another important tool that's used in monitoring the activity of organisms and ecosystems is PCR, polymerase chain reaction. So what I'd like you to do is to watch a portion of a module that we prepared for Biology 101 that will explain PCR to you. Okay, today I'd like to continue our discussion of DNA technology and talking about some different examples of technology and how they're utilized. So the first one I want to discuss is something called polymerase chain reaction, or PCR. And the purpose of PCR is to make copies of a gene. And so it's a pretty simple process in that it involves a cycle of heating followed by cooling. And then these alternate over and over again to produce our copies of genes. So it's in that heating and cooling cycle, it's a three-step process. And so I'll start with this, then we'll look at some diagrams. But the steps are denaturation, and so denaturation will occur at the peak temperature, about 94 to 96 degrees, and this causes the separation of the two strands of DNA of interest to us. And then in the cooling phase, there's initially what's known as annealing, where a short segment of RNA will recombine with the strands of DNA. And then extension would be where DNA polymerase then extends each strand of DNA to get us back to a two-stranded piece of DNA. So let's go ahead and look at some diagrams that kind of illustrate this for us. But before we get to that, what we would do then in this PCR process is that the PCR cycler basically then is like an oven. It will heat and cool. And so in one of the tiny vials that we utilized in our last module, what you'd do is put some sample of DNA, and that DNA sample then would have the gene of interest to us. And one of the good things about PCR is that we don't need to know exactly where those genes are, but what we do need to know is some of the sequences on either side of our gene, and we'll see that in a couple of seconds. So anyway, your DNA sample of interest would have to go in this little vial, and then the nucleotides that we would need to build new DNA strands. And of course, our 4 nucleotides would contain adenine, guanine, cytosine, and thymine if we were making DNA. And then we'll also need some short RNA primers, and we'll see why we need those in a second. And then also a type of polymerase that we call Taq polymerase. And Taq polymerase is a heat-tolerant DNA polymerase. It comes from a heat-tolerant bacteria known as Thermus aquaticus, and that's where the Taq part of this comes from. So again, it's a DNA polymerase that tends to work at higher temperatures than DNA polymerase normally does. Now, in the process itself then, what we would start is with a chromosome. And of course, this is not to scale. The chromosomes are really long. But we would be interested in a particular gene in this chromosome. And so we'd be targeting a particular gene. But again, what's important about PCR isn't so much where that gene is, but what we would need to know is some of the sequences on either side of the gene. Okay, and I think that'll become obvious here in a second. So anyway, again, we go through the heating cycle in which the DNA double helix will separate and the strands separate. So the denaturing of the chromosomes will occur, and again, this is separation of the DNA strands. Then in the cooling phase, initially what would happen is that RNA primers, short pieces of RNA, would reanneal with complementary bases. And so again, this is why, even though we may not know everything we want to know about the gene, we do need to know some of the sequences on either side of our gene, because we have to produce primers that are complementary to the bases on either side of our gene. And then they will anneal with those strands of DNA. Now, this is an important step because you may recall that DNA polymerase cannot bind to a single strand of DNA. So again, what we want to do here is produce complementary strands of DNA so that we end up with 2 copies of our chromosome or 2 copies of our gene as opposed to having 1 copy. So DNA polymerase requires a double strand to bind to, so that's the purpose of the RNA primers. We need the primers there. They will anneal with the complementary strands. That then allows DNA polymerase to bind to those double strands. The DNA polymerase can then build new strands 5 to 3, and at the end of the process then we will have 2 copies of our gene at the end of a 1 heating and 1 cooling cycle. And then again, it's a very simple process then, so still leaving that stuff in the vial then, we just need to go ahead and continue the heating and cooling cycles to make more copies of our genes. And so now we've got 2 copies of our genes, we'll heat and cool again. So the heating process will cause denaturization. In the cooling cycle, we'll get reannealing. the DNA polymerase will bind, and then extension of the strands. And that will double our copies of genes again. So what happens then in this process is that with each heating and cooling cycle, we effectively will be doubling the number of copies of genes that we have. And again, there's some important uses to this, but you might be wondering, why do we need a lot of copies of genes? And I'll get to that a little bit later. So we do have some situations where we would want to use multiple copies of genes. Now, one of the applications of PCR is that we can actually use this in concert with gel electrophoresis to produce DNA fingerprints. And so this is especially useful when you don't have a big sample of DNA, when you have just a small amount of DNA, and you might not be able to run a traditional DNA fingerprint, as we discussed in the previous module. So what you can do in those situations then is that you can, again, target a particular gene. We can run PCR, but what we would be targeting here is not just any gene. What we'd be interested in targeting would be VNTRs. So what we could do then is target specific VNTRs, and then produce primers on either side of those VNTRs, and we could make multiple copies of those VNTRs, specific VNTRs. And then, again, if we target several different VNTRs, what we would do is be producing PCR fragments instead of restriction fragments, if you will. So then we could go ahead and load our samples into a gel. We could go ahead and run our gel. And again, the nice thing about this is that when we run a traditional DNA fingerprint using the restriction enzyme kind of techniques, you end up with thousands of different bands. But in PCR, you'd be amplifying specific genes rather than thousands of genes. You'd just be targeting specific genes. And so anyway, it can make for a simpler type of amplification, and you won't necessarily have as many bands. And in some cases, again, you don't even necessarily need to use probes in some of these situations. It just kind of simplifies the whole process. But probably the best use of the PCR, again, in concert to produce the DNA fingerprint, is that you only need tiny amounts of DNA. You can amplify specific VNTRs, and it works great. So a lot of times these are used together.